Hello, I'm Johnathon Richardson from Humboldt in the Department of Fisheries Biology. My advisor is Andrew P. Kinzinger, and I'll be discussing my thesis research work on the dynamic fluctuations in fish communities of Humboldt Bay. It's a seasonal analysis using environmental DNA metabarcoding. Now, some of you might be asking, what is environmental DNA? So environmental DNA is DNA that's been extracted from environmental samples rather than the organisms themselves, where it's like sloughed off and shed into the environment from these organisms. In particular, my samples were collected from water from Humboldt Bay and then extracting this environmental DNA. And from this, I'm able to find out what species of fish are living in this community of Humboldt Bay over time and see how as the year progresses, how these communities fluctuate. So as I mentioned earlier, these are derived from either epidermal cells of sloughed off feces or urine from these organisms, and that I get this mixture of eDNA from the environment when I collect these water samples. It does degrade into smaller fragments, which I have to use software to then reconstruct and analyze. But these eDNA samples in the environment can persist for a couple of weeks, depending on environmental conditions such as UV light, oxygen, salinity, and temperature of the water at that time. Now, some of the uses for eDNA for species monitoring is detection, site occupancy. So since I'm using water, I can go and scoop out, collect a sample of water, and then analyze that to get the inventory of the species in that area. And it's quite sensitive, and so from that I have to build a custom reference database to filter out some other non-target organisms I don't want to look at, such as marine bacteria or even things such as mammals or birds, and just focus solely on fish species. Now, there is still some work being done of seeing whether or not you can use environmental DNA to estimate an abundance of some of these species. Unfortunately, at this time, it's still research ongoing. This is more of an occupancy of a presence absence for these species. Now, some of the advantages of environmental DNA for species detection is sensitivity. So for environmental DNA, it is highly sensitive. So for traditional trawling or sampling methods, there's a higher likelihood of not encountering the species, whereas with environmental DNA, it's quite a bit higher. For example, for environmental DNA, we were able to detect tidewater goby in the Klamath River that was not previously sampled or caught with traditional sampling methods. Another thing is it's non-invasive. So there's no take or handling of the targets. There's minimal disturbance to the habitat. I'm not dragging a net over a benthic community and disturbing the ecosystem. I'm just going out there and collecting a 2-liter bottle of water. And then finally, reducing permitting. So I just need a field collection permit to go collect it. It removes a lot of the red tape involved. There's no animal care permit required since I won't be handling any of the animals. I'll just need the access permit to access the site. So for example, if you wanted to find out the fish community underneath the Golden Gate Bridge in the San Francisco Bay, you won't have to deal with all that litigation or like red tape to go out there and just collect your cup of water, because people aren't too concerned with collecting water. So like I said, I go out there, I have a 2-liter sample, I collect in triplicates, so there's a total of 3 of them plus a field blank, the water. It's basically attaching a bag to the end of a fishing pole and hucking it into the bay about 5 to 15 meters. And from there, I take it back to the lab to do the filtration. And I pass the water samples over a filter pad that is a 0.45 microns, and it helps filter out a lot of the other sediment and other material from there. And then finally, I do the extraction process where I use a Kyogen kit, and that includes a lot of buffers and shredders and spin columns to help distill my samples even further. And then finally from there, I'm able to digitize the samples into a thing called FASTQ files using next generation sequencing that produces tens of thousands of DNA sequence reads. And then by using bioinformatics, I'm able to then document the fish biodiversity and make an inventory of that. So as you can see, I was able to make a little map here. This is the Sequoia Conference Center. The samples I took are not very far from here. They're at the Samoa boat ramp, for those who are familiar. It's just a little, almost 7 miles away. Let's see. And here's a little picture of what the site looks like. It's really readily accessible. It's a great reference point for collecting the bay. It's also very lenient as far as weather goes. I can go out there when it's downpouring, when tide is high. I'm still able to go out there and collect my samples on a bi-weekly basis. And from there, after doing the bioinformatics and analyzing it, I was able to find a total of 109 species thus far for the Humboldt Bay. And when this is compared to traditional sampling methods that have taken place over the past 15 or actually 50 years, many of the traditional field collections weren't able to quite reach that same number. And there's due to reasons of that. For example, we're dealing with a lot of like benthic, cryptic, small species that live in rockwork, where if you're trying to trawl a net through, you'll end up just damaging or losing your equipment. And so from using environmental DNA, we're able to get a more clear picture of the biodiversity and communities of these fish that are living in the bay. And this is a little hard to read for some of you, even some in the front, really. But the gist of this is I have a nice little, on the y-axis, I have a list of species from highest abundance of read counts. So like the most little sections of DNA that eDNA was able to collect to least. And on the bottom of the x-axis, I have a date starting from February of 2023 through December. And the circles, the different sizes are like little salmon ones are like small reads between like, at smaller end, green's middle, and blue is large. There's tons of those reads. And so if we look at the top, or I can look at the top, the one that really jumps out is like Pacific herring. It's ubiquitous in the bay. It's commercially collected. It's very common. But as you continue down the list, you start coming across more uncommon and then rare and then rarely seen species. And then from there, you can kind of build a map of like, okay, when do we expect some of these species to start moving into the bay? Next, from analyzing that further, there's a couple alpha diversities I was able to generate, observe the Chow one and then Shannon, to kind of see how this changes over time. Now, I have 3 points for each of my triplicates for each of the dates. And the alpha diversity is like how many species you should expect to observe for that sampling. But you can see how it fluctuates as time goes on this slide here. And now, it's interesting thing on this one with the February, it was one of the earlier ones, it looks relatively low. But the reasoning for that is we suspect that the Pacific herring comes into the bay to spawn at that time. And so they're releasing a lot of their like gametes into the water, which then once that's collected in the water sample, there was a bias in the PCR process of replicating those samples where you're more often not going to run into a Pacific herring sample and make more of it. So they are overrepresented and they're overshadowing a lot of the other species. But once they, moved back out of the bay, you see that alpha diversity resumes to a more of an expected amount. So looking at that list, some of the commonly detected species in Humboldt Bay, we of course have the Pacific herring. We also got a very high recounts of the kelp greenling. We have black rockfish. And another one that was very interesting was the monkey-faced prickleback. So on traditional sampling methods, they don't really end up all that often in your nets. But with using environmental DNA, this more cryptic species that lives more in like the rock work of the jetty is more accurately represented because environmental DNA doesn't care about or is stopped by the rock work. And so this fish that's normally collected using poke poles, you're able to more accurately see its abundance or more of like persistence in the bay. Moving forward, we have a couple of more occasionally detected species in Humboldt Bay. We have the rosy-lipped sculpin, we have the California halibut, we have the red Irish lord, we have northern clingfish, we have the petrale sole, and of course we have the lingcod. Now some of these species have been caught regularly through traditional methods, but eDNA, they were quite regularly detected in the systems. And then finally, we have the rarely detected species in Humboldt Bay. So we had a couple of hits of the king of the salmon. We have which is a more oceanic fish. So I guess a sample of it came in with the tide. That one, or those few sampling periods we had. We have the black-eyed goby, which is a very small benthic fish, which is more difficult to collect. And then we have the sail-finned sculpin, which it was suspected to be in the bay, but wasn't really seen in these conventional methods. And then, of course, we have some other rarer species, as like a painted greenling, a Pacific butterfish, and I think that's the hole in the head sculpin, another one of those rocky, small, cryptic species. And then we also had some very extremely rare detections in Humboldt Bay, which you wouldn't really suspect. So we have the white croaker normally from Southern California, from its range. We had a couple detections of that. We also had a Kamchatka flounder, which usually for this range you would expect more of the arrow tooth, which looks a lot like it. But I had both in my reference database, but it always referred to the Kamchatka. And then finally, we have the broomtail grouper, which is a Southern California species. We had one detection of it, and while it could have been one individual that swam up with a little vagrant visit, it could also be with environmental DNA. You're not able to tell the life stages or the sources for a lot of these detections. So it could have been maybe somebody sailed up from Southern California, had some of this grouper on their deck and was rinsing off their boat. It somehow ended up in the bay, washed over to like the Samoa boat ramp, and I got picked up. And then finally, with some of the threatened species, we detected a long-fin smelt, the green sturgeon, and of course the steelhead and chinook as well in the bay. An interesting thing with the exotic species, so we have the ones that are usually planted like American shad, which had a couple, as well as the striped bass. But an interesting one was 6 detections with very high, or relatively high recounts of the yellowfin goby, which hasn't been officially documented in Humboldt Bay, but it looks like it's been consistently popping up in my samples that they have a presence now in the bay. In conclusion, this is eDNA is an effective technique for recording fish diversity of Humboldt Bay. Detected quite as many species as conventional monitoring of trawling, and seasonal variations with this fish diversity was documented. I want to thank DR Reeds and Associates, and of course the U.S. Army Corps of Engineers, and also those who have provided assistance in collecting this research. And do any of you have any questions?